Okay, so in class, what we did was we talked a little bit about gel electrophoresis and how scientists use this to study particular fragments of DNA after they treat them with restriction enzymes, and they can use it to answer, a variety of types of questions, from crime scenes to paternity suits, etc. So agarose gel is typically what's going to be used, and you're going to put this into an electric field, and then you're going to add the DNA fragments to it into the wells, as you can see here. This is kind of like what we went over in class. You're going to add the DNA into these particular wells, and then DNA, being negatively charged, will move towards the positive pole, and it's going to be separated based on size. The shorter molecules are going to move a further distance than the larger molecules will, and then you can stain this a particular way. This was stained with ethidium bromide here, put under a UV light, and then what you can see are the different fragment bands that are there. Okay, so DNA, as we said, is negatively charged. It's going to move towards that positive pole in the electrophoresis box, and treating it with a variety of different types of restriction enzymes is going to give you a way to really manipulate the DNA in a way that's going to give you a lot of information. Okay, so a lot of DNA contains these short sequences that repeat in a typical pattern, like AAATTG, AAATTG, and so forth. So it's going to continually repeat. And there's a variety of different types of repeats, but what these are called is short tandem repeats, and they're very useful because what they do is they can give you information about DNA in terms of like a fingerprint. So everybody has a unique banding pattern that they produce from these tandem repeats. And these polymorphisms, there's all sorts of different types of forms within the chromosomal DNA that can arise within these repeats. And the number and type of repeat that you have varies from person to person, but it's unique to you, just like we said your fingerprint is. So it serves as a molecular marker that can provide a lot of useful information for like paternity, crime scene, etc. Okay, so what you do is you use a particular restriction enzyme to cut the DNA sample that you have at particular sites, and you're going to get DNA fragments of varying lengths depending on how many repeats there are within that particular fragment. And the variation in the particular number here leads to the differences that you see in the individuals. So your pattern is going to be different than mine. It's going to be slightly different than our parents, but it's going to be more similar to our parents than it is to us, unless, of course, we're siblings. Okay, so these tandem repeats give you a lot of information. So what you're going to do is you're going to analyze these restriction fragments, as they're called, because you've got these fragments of DNA. You've treated them with restriction enzymes, and you start to produce a bunch of different little smaller and smaller fragments. And this is what scientists can use to then analyze the differences in those DNA molecules. So you treat the DNA, of course, with a restriction enzyme. You run the samples through a gel, and then you get a banding pattern that is characteristic to the particular person that you're pulling that DNA from. And then this can be used to examine the presence of certain genes and bacterial chromosomes. You can settle a paternity suit. You can provide evidence to prosecutors from a crime scene standpoint, and it gives you a whole host of information. So then when you have these non-coding regions of DNA that you're treating with these various restriction enzymes, this is where the scientists have discovered the differences in these non-coding regions on the homologous chromosomes. So what they were given the name of is a restriction fragment-like polymorphism. So in other words, the differences in these non-coding regions is what provides for the genetic fingerprint. So this would almost be like the intronic DNA. You could think of, there's long stretches of DNA within your chromosomes that don't code for anything. And then when you treat the DNA to isolate these fragments, that's when you're going to start looking for the repeats. Once you've got those repeats there, you can treat them with certain other restriction enzymes and kind of break up those repeats based on where you're cutting them. And then the number of repeats that you have, the number of different fragments that you'll generate, different sizes that you get, that's what's going to provide for the particular genetic fingerprint that you're actually looking for. And what these restriction fragment polymorphisms kind of serve the purpose of is a genetic marker, and it's within that non-coding region of the DNA. And what it does is it's very near a particular locus for an allele or a gene within that genome. So there's a lot of different variants within a particular population, but these differences within each person are enough to really kind of mathematically make it impossible for it to be someone else. So in other words, if they find your DNA at a crime scene, or if your fragment pattern matches that, within like a child, as we went over in class when you're looking at the different restriction fragments, the chances that it belongs to somebody else, in other words, the chances that you and somebody else have the same restriction fragment length polymorphism pattern, is vanishingly small at best. So in other words, it's a pretty kind of rock solid way to identify whether or not the person committed the crime, whether or not they're the father of the child, for instance, etc. So there's a variety of different types of things that it can be used to do. So when you're analyzing these things, what scientists typically do, again, is they take the DNA and they treat it with a restriction enzyme, and then they run it out on a gel electrophoresis. And then they do this technique known as southern blotting. And what you basically have is you have a solution that's going to pull out particular fragments of DNA, or it's going to pull out the fragments of DNA rather. And what it does is it wicks them up and puts them on a piece of nitrocellulose paper. So you've got a heavy weight here that's pressing down on this entire thing, and basically you've got this sandwich of stuff, and as the solution is kind of pulled up through the setup here, the DNA gets stuck onto this nitrocellulose paper. Okay, and then once you have the nitrocellulose paper, you can put it into another kind of preparation here, just really a plastic Ziploc baggie, and then you can put these radioactively labeled kind of probes that are designed to test for the presence of particular pieces of DNA, particular fragments. You can put them, mix them in with this entire thing, and what these things are going to do then is bind to the fragments that they match up with from this gel, which are contained on the nitrocellulose paper. So mixing them in, they'll bind wherever you need them. And now you've got this piece of nitrocellulose paper that you can put with a piece of film, and then that radioactivity is going to develop the film in the particular spots where these probes have bound. And again, they're binding to the particular patterns that was in the DNA that got stuck to that paper. So what you have now is this film that's providing you with a particular genetic pattern, a particular DNA fingerprinting pattern that will then kind of reveal the information about the person or the gene or whatever it is that you're after. So for instance, this film right here was treated in the way that I had just mentioned to you. And so if you look at this, what you see is you've got the defendant here, and then you've got the victim. And on the defendant's jeans and on the defendant's shirt, there was some samples of blood. And the defendant can give you a variety of different ways in which, reasons there's blood on the clothes that he was wearing or she was wearing. And you can look at the victim's blood. And when you do that, the question becomes, why does the victim's blood appear on the defendant's shirt and jeans? And the defendant can say, well, it's my blood. But when you start looking at it, again, we had mentioned here, for instance, that sometimes we share these patterns with all of the, or with other individuals. But if you look here, it's not here, okay, or here, it's not here, or here, etc. But these three, the victim blood is on the defendant's clothes. And now the question becomes why? And you can use this for paternity suits the same way, but this is some pretty sound evidence that somehow the victim's blood got on the defendant's clothes, and the defendant doesn't really have a valid reason for why that is. And then you can see that this is some evidence that will then kind of hopefully lead to a conviction of the defendant. Okay. The other way that it can be used is to find out if you have a particular gene. So in this particular case, you can treat the hemoglobin gene with a particular restriction enzyme, and it's going to cut at these 4 spots if the gene is normal. And then if the gene is a mutated version of it, it's only going to cut in these 3 spots. So what you'll get here is you're going to get 1, 2, 3 fragments from the different cut sites in the normal hemoglobin, and you're going to get 1, 2 fragments in the mutated version. So what you can do is use this to kind of confirm whether or not a person has a particular gene or mutated version of the gene, disease, disorder, and you can plate it out on the electrophoresis and look for the banding patterns. So if you have the normal hemoglobin gene and you do not have sickle cell anemia in this case, then you're going to get 3 fragments. And then if you do have the mutated version, you'll get 2 fragments, and that would be a way to confirm that the person might have sickle cell anemia, for instance. Okay, so there's a variety of diagnostic things that you can use for this as well. Other things that it's used for is sometimes there are certain RFLP restriction fragment length polymorphism patterns associated with a particular disease. So what they'll do is they'll look at and see the banding pattern here, and if you've got two of these markers present, then there's a real good chance that you might have the particular disease-causing allele. Whereas if you only have one, then what that might say is you might have the normal allele. Okay, and again, nothing here is 100%, but again, you start looking at the probabilities of it, and then that's what's going to give you the information that you're after. Okay, so that takes care of the use of restriction enzymes to create these fragments and banding patterns to answer a variety of different questions that scientists and people in the legal community, molecular biologists, etc., what they might use it for. So if you have any questions, of course, make sure you ask. I know a lot of this stuff can be very difficult to understand, and if you have questions, we always have time to go over these in class. All right, so we'll talk to you soon.